Welcome back, biologists. We are in the middle of chapter 36. We've just discussed how a plant can absorb some of the nutrients it needs and how a plant can perform photosynthesis to create some of the nutrients it needs. But now that you have those minerals in the roots or that sugar in the leaves, how is a plant going to get it to where it needs to go? So there's a few different paths that can be taken for things to diffuse from cell to cell. One is by moving outside of cell membranes, and one is moving inside of cell membranes. So the apoplast refers to everything outside of the plasma membrane. Here's a plasma membrane, here's a plasma membrane, here's a plasma membrane. This apoplast would be cell walls, extracellular spaces. It even includes interior of dead cells. So those dead cells would be things like vessel elements and tracheids of vascular tissue. That's the outside the cell membrane way. So if something is going through an apoplastic route, it would be water traveling from cell wall, inner membranes or extramembrane spaces. The symplast would be the inside of the plasma membrane. So what's in the cytosol? So a plant nutrient would get into the cell membrane and then travel through these plasmodesmata. Those plasmodesmata are those little junctions that connect the cells to each other and just go from cell to cell to cell to cell. That's the symplastic route. There's also one more transport route called the transmembrane route, which kind of goes through a little bit of apoplastic and a little bit of symplastic. It just can't make up its mind. So it gets into the cell membrane, travels through the cell membrane, through the inside of the cell. Then it goes back out, leaves the cell, travels through the cell wall, back into the membrane, through the cytosol, back out to the cell wall, back into the membrane. So this transmembrane route is going to require going across the membrane, which is why it's called transmembrane, repeatedly crossing the membrane to go from one cell to the next. This type of short distance transport of solutes is nice on a cellular level. The plasma membrane permeability will control what's able to come in and out. Some things are able to just diffuse right through that cell membrane. There's also various types of transport proteins. Here's one like a proton pump, pumping hydrogens out. Those transport proteins are involved in moving things across the cell membrane to allow them to enter one of those transport routes. Water can also go through a cell. We spent a lot of time last semester talking about osmosis. Here is the technical description of how you can tell where water moves. I find it easier to say water will follow the solute. So wherever there is more salt, wherever there is more sugar, wherever there is more stuff, wherever there's more concentration of stuff, water is going to move towards that. So if you have a cell in a hypotonic condition, hypo means low, there's not a lot of solutes around it. That means there's going to be more stuff in the cell. Water is going to move into the cell, and that plant will start to swell, and you'll end up with a turgid cell. However, if there is a lot of solutes outside of the cell, a hypertonic, hyper meaning high, there's a high level of solutes around the cell, the water is going to move out of the cell into the environment. That means that plasma membrane is going to shrink, and you'll end up with a wilted plant. So solute potential just means osmotic potential, and that is just controlled by the solute concentration. There's also pressure potential. Pressure potential is the physical pressure on a solution. So how much pressure is inside of the cell compared to the atmosphere. Water is going to move from positive pressure to negative pressure. So think of like if you're squeezing a balloon, you're putting pressure on that, so water will come out of that water balloon. Water in living tissues is usually positive pressure, and dead hollow tissues is usually negative pressure. The difference in water potential will determine the movement of water in a plant. There's also, not only will that control where the water is, which direction the movement is moving, but there are also things called aquaporins that control how much water is moving. So we see much more water movement across a membrane than can just be accounted for by diffusion because of these aquaporins. These aquaporins are channels that allow water to cross right through the cell membrane. So they facilitate osmosis. They can also open and close in response to cell signaling. So if there's more calcium inside the cell, they'll close. If the pH decreases, they'll open to allow water to move. Sorry, that captioning kind of has me a little bit distracted. If the pH decreases, they will close. They will open. Okay, let me focus now. Okay, however, diffusion is not going to move water very far. If you waited for diffusion to take water from the roots up to the top of a tree, you'd be waiting for centuries and that plant would be long dead. So there is something called bulk flow that will more effectively take water over long distances through that plant. Bulk flow is regulated more by pressure gradients. So fluids flowing from high pressure to low pressure, independent of the concentration of solutes around them. Bulk flow primarily occurs in xylem and phloem of vascular tissues. This would be the tracheids and vessel elements of the xylem, and the cytoplasm of the sieve tube elements. And those sieve tube elements in the phloem is almost always devoid of organelles. So that ensures a larger diameter in the vascular tissues so you can have increased and more efficient flow. All right, at this point, you should be able to tell me, how would a reduction in the number of aquaporin channels affect a plant's ability to adjust to new osmotic conditions? And how would long-distance transport of water be affected if tracheids and vessel elements were alive at maturity? And what would happen if you put plant cells in pure water? All right, those should be questions you can go back, hopefully answer from memory of just participating in this lecture video. Or if you need to, go back and review your notes, reread your textbook, and I'm always available during office hours. And I will see you soon for our next video. We're going to talk a little bit more about how flow is happening in our xylem and phloem. I'll see you later. Bye.